Welcome grade 12. I want to welcome you to the Junior Tukkie Winter School. There will be a series of videos which will be live streamed in the next few weeks on the juniortukkie.online platform. Please use the given material as revision and also look at all the grade 12 content for life sciences on our platform. I am Miss Kutsia, and today we will look at the Life Sciences Paper 2 that was written in November 2021. So grade 12s, we now move on to the last question, which is question number 3. So question number 3 will also form part of section B. So question 3 is 50 marks of the 100 marks that's allocated to section B. So in previous years, there was a section C with an essay question, but that was removed. So we only have 2 sections. So let's look at question 3.1. So here was no diagram given. As I looked at this image, I at first thought that this was regarding genetics, which was a bit confusing because there was quite a lot of genetic questions that was asked in question 2 already. But as you read through this section, you will notice that they are just identifying favorable traits that they have noticed in horses. So they say some horses have straight hair and others have curly hair. A scientist wanted to clone a straight-haired male horse to meet the demands for horses with straight hair. So here the clue was that this section was about cloning. So this is artificial selection. So there's a high demand from people for horses with straight hair, and the scientist wanted to take a shortcut to make sure that they have the exact trait that is demanded for, and they wanted to clone the horse with straight hair. So before we read through the entire question to examine every single detail of the question, let's go and have a look at what cloning is. So here I made a very short summary on what cloning is and how cloning is done. So the first animal that was cloned was known as Dolly the sheep. She was the first animal that was cloned. Currently, it's illegal to clone humans. There's quite a lot of research that still has to take place behind cloning. So what they did was they took Dolly the sheep and then they took a second sheep. You will see Dolly the sheep was a plain white sheep, where the second sheep was quite darker. Let's for instance say call it the grey sheep. So what they did was they took a somatic cell from Dolly. So what a somatic cell is that it is a body cell. And what is very important about this is that it contains 46 chromosomes. It's not the ovum from Dolly. It is a body cell containing all the genetic material of Dolly, not just half of it. Then they took the ovum from the grey sheep. And what they did was they removed the nucleus from this ovum. The reason for it is because I want to remove the genetics of the grey sheep, because my goal is to clone Dolly the sheep. So what they then did was that they used electric shock method to fuse these two cells. So they fused the somatic cell of Dolly with the ovum of the grey sheep. Then what they did was they took this new cell and they implanted it into the uterus of the grey sheep. And after a few months, they had a small little lamb called Dolly. So this small lamb will be an exact clone of Dolly the sheep. The downfall with this was that Dolly was already 6 years old when she was cloned. So technically, the cells that made up the newborn lamb was already 6 years old. So that meant that this lamb did not survive for a very long period of time. So let's read and see what is expected from us in this question. So the first sentence we have read before, so let's quickly go through it again. So some horses have straight hair and others have curly hair. So a scientist wanted to clone a straight-haired male horse to meet the demands of the horses with straight hair. So the scientists used the following procedure. So exactly the same procedure which I explained now with Dolly the sheep, they did with this male straight-haired horse. So they said the nucleus of the somatic cell was taken from the straight-haired male horse, which they call horse S, and an unfertilized ovum was removed from a curly-haired female horse, which is known as horse T. What they then did was the nucleus from the somatic cell was then placed inside of the ovum of horse T, and then the ovum was then placed into the uterus of the female surrogate horse, that is horse R. So we had horse S, which was the male straight-haired horse, then T, or horse T, which is the curly-haired female horse, and then horse R, which is the surrogate. So this will be the horse that will carry the foal. Question 3.1.1 states that they want you to explain why a somatic cell and not a sperm cell from horse S would provide the nucleus for the procedure. So it's so important to note that horse S was the horse that we are trying to clone. Then question 3.1.2. So before we insert the nucleus from the somatic cell of horse S, the nucleus from the ovum of horse T has to be removed. So they want to know, why is this procedure so important? The word significance means what is the importance of this statement or this procedure. Question 3.1.3. To which of the three horses, so horse S, which is the one that we are cloning, horse T, the horse that we use the ovum of, and then horse R, which is the surrogate, will the cloned offspring be genetically identical to? So they want to know, out of the three horses, which one is the clone genetically identical to? Then question 3.1.4, you need to state two benefits of cloning. So the word benefit means why, or two importance, or why is cloning important to us? How can we benefit from cloning? So let's look at the marking guidelines. So question 3.1.1 said that you have to explain why did we use a somatic cell and not a sperm cell from the horse that's being cloned. So firstly, the nucleus of the somatic cell is diploid. So that means it has a full set of chromosomes. And the nucleus of the sperm cell is haploid, which means that it contains half the sets or the half the amount of the chromosomes. So the somatic cell will then carry the desirable characteristics, and it will contain the whole DNA strand. Question 3.1.2. So before inserting the nucleus of the somatic cell into the ovum, why was it important for us to remove the nucleus from the ovum? So the reason for this is so that we can ensure that the DNA of the ovum or the characteristic of curly hair is removed. Because remember, the ovum that was used came from a horse with curly hair. And this is not the desirable trait. So this is not the trait that's in demand. So we had to remove the genetic material that might carry the characteristic for curly hair. So the reason why then is we only need the DNA from the somatic cell from the clone, because we only want the amount, or we only want the allele for straight hair. Then also we want the correct number of chromosomes in the clone. So for example, if this was human, we want 46 chromosomes in the offspring, in the zygote that's going to form, not just 23. Then question 3.1.3, they want to know which of the three horses will the cloned offspring be genetically identical to, and that would be horse S, because this is the horse that we wanted to clone. 3.1.4 state two benefits of cloning. So to produce offspring with a desirable trait, to conserve certain threatened species or endangered species, or what we can do is to create tissues or organs for organ transplants. And for this question, we will only mark the first two.
In question 3.2, we will look at the present-day distribution of three closely related species of the dog family. So here we will look at the coyote, the jackal, and the dingo. And this is shown in the map below. So here you will notice that they will indicate on the map how these species are distributed across the world. So here it will give us an indication that once these species were living in a very close area together, and as they got separated, they will then obviously change into different species depending on the environment that they were exposed to. So let's quickly look at the summary of what is happening in this question.
So let's have a look at geographic isolation. So in the question, they asked us what is a biological species. So a species is a group of organisms that is allowed to interbreed and produce fertile offspring. So in order for them to be categorized as a species, you need to produce fertile offspring. For example, if we take the horse and a donkey, they can interbreed with one another, but then they produce an offspring that's infertile, and we know that as a mule. So what happens in geographic isolation is that this specific species was separated by a geological barrier. In this case, it could have been a river, an ocean, or even a mountain. So what it caused was that there was no gene flow that took place between these two species, and they cannot interbreed with one another. So due to the different environments that these species were exposed to, the organisms with a desirable trait survived in this new environment. And this caused a change in the species, which caused the development into different genotypical and phenotypical differences. So this caused that changes. So this caused the species to be genetically different from one another and also phenotypically different from one another. So question 3.3 has to do with a graph. So before you read anything, it is so important for you to understand what goes on in a graph and what the graph is telling you, because it tells you what is happening, what this experiment is about, and then exactly what the conclusion is going to be. It is obviously going to tell you what happened in this experiment. So if you do not understand the graph, there will be a very small percentage chance that you will be able to answer this question. So let's read through the question, and then I'll explain to you what this graph is saying and exactly what is happening in the graph. So in this case, they gave you a graph instead of asking you to draw a graph, but if they give you a graph or the table, the same conclusion can be made from the table and from the graph. So they say some farmers add low doses of antibiotics to the feed of cattle. The use of antibiotics in cattle feed could result in the evolution of antibiotic-resistant bacteria. So, number one, we know that antibiotics is a medication that's used to kill bacteria. So these farmers will give very low dosage to the cattle every day in their feed so that we can manipulate the evolution of bacteria, especially for the antibiotic resistance. So in this graph, the graph will show you the effect of different doses of antibiotics on the number of harmful bacteria in cattle. So if we look at the heading of my graph, it says the effect of the different doses of antibiotics on the number of harmful bacteria in livestock. So in this heading, they mention your dependent and your independent variable. So the different doses of antibiotics, that will be your independent variable, because that's the doses, the amount of antibiotics in milligrams that the farmer chose to add to the feed of the cattle. And then what is measured in this graph will be my dependent variable, and that will be the number of harmful bacteria, not just bacteria. What about the bacteria? The number of harmful bacteria. So on your y-axis, you guys will notice that it gives you the number of harmful bacteria. And then on your x-axis, it gives you the antibiotic doses in milligrams. And then they gave us a simple line graph, which is one of my favorite graphs because it cuts to the chase and it tells you exactly what happens. So you will see at a very low dosage of antibiotics, we have a very large number of bacteria that survives. And then as I increase the dosage of antibiotics, there will be a decrease in the number of harmful bacteria. So this tells me if I increase the antibiotic dosage, if I increase the amount of dosage of the antibiotics, it will kill more harmful bacteria than a lower dosage. And there you already have your conclusion if you can figure out what this graph is about. So question 3.3.1 asks, use evidence from the graph to explain why higher doses of antibiotics will benefit the farmer economically. And then question number 3.2.2, explain how the use of antibiotics in animal feeds may result in the evolution of antibiotic resistance. So let's look at our marking guidelines. So if we look at question 3.3.1, they asked us to use the evidence from the graph to explain why higher doses of antibiotics will benefit the farmer economically. So it will decrease the number of harmful bacteria, so it's going to kill them, because you can see on the graph, the higher my dosage, so if you see if we move from 2 to 10, there will be a decrease in the number of bacteria because the line graph is moving downwards. So it moved from, if we look at 2, if we move up, it moves from 1,000. If we move down to 10, it moved down to about 200. So we will see that the number of bacteria that survived is much, much lower in a higher doses. So therefore, we prevent the cattle from getting the disease or being penetrated by the pathogen. Then also it will decrease the mortality rate of the cattle. So obviously a larger amount of cattle will survive for the farmer then to sell or to breed or then to increase his profits. Then question 3.3.2 says, explain how the use of antibiotics in animal feed may result in the evolution of antibiotic resistance. Okay, so firstly what happens here is it's due to natural selection. So we are bargaining on natural selection, in this case, to use the antibiotic resistance to our advantage. So natural selection occurs because there's variation amongst the bacteria, and then some of the bacteria are resistant to the antibiotics where others are not resistant. So when antibiotics are added to the animal feed, some of the bacteria that's not resistant will obviously be killed, they will die, and those that are resistant will survive and then reproduce. So then this characteristic that is resistant against the antibiotics will then be passed on to the offspring. And then the next generation will have a higher proportion of antibiotic resistance. So as I read through question 3.3.2, I actually thought to myself, then why would farmers actually make use of antibiotics in feed? Because if I look at the graph, it makes sense to me why they would do it. Because
they just increase the dosage to a certain amount, and then if the cattle ingest it, it actually kills the harmful bacteria, and it is a way of preventing this bacteria of making your animal sick. And obviously we know that veterinaries cost quite a lot of money because you have to get them out to the farm and pay a certain amount to get the vet to the farm, and then you actually have to pay the vet to treat the animal as well, which is quite expensive. So I do understand that it helps the farmer save a lot of money. But if you then look at question 3.3.2's answer, where we explain how resistance builds up in these bacteria, we see that this is not a very effective way of treating bacteria, because this in the long run will have a massive effect on our livestock, because the harmful bacteria will become resistant, and then that antibiotic will no longer be effective against this bacteria, and then we will end up losing more cattle, and it's going to cost that farmer quite a lot. So in the long run, this is definitely not effective. But there's certain guidelines that is put in place by the FDA. So this is your Food and Drug Administration. So there's certain bylaws that these farmers have to follow for us to then, or for them to make sure that this antibiotic resistant bacteria evolving into this antibiotic resistance bacteria, so that we can prevent this for much longer, if I can put it like that. We want to delay the resistance actually. So it is an effective way. It does work because it ensures that we get healthy animals, and obviously we can then produce the meat much faster in that way. So it's actually not a very effective way in the long run, as we then produce antibiotic resistant bacteria. Question 3.4, the famous investigative question. So in question paper 1 or in question paper 2, does not matter, from grade 10 to grade 12, you will have one investigative question with very, very similar questions that would be asked in things like this. The only difference is the scenario that will be asked. So in this case, it's about a female gull fly. So what makes this very difficult to understand is because most of us do not know what a gull fly is, because it is quite difficult to understand a new concept, because as you're sitting there in the exam, it would be most likely the first time ever that you have heard of a gull fly and what is happening in nature. So let's quickly just read through the question at the top, the information that's been given, and then we'll go through the summary, the things that you need to know that's very important for an investigative question. So just remember, I'm giving you the summary. I'm not giving you the whole situation or everything that you need to know about an investigative question. Here you can go back on information that's been posted on the grade 12 platform. So they say that the female gull fly lays eggs on the stem of plants. So the eggs will hatch to form larvae that secretes a substance into the plant tissue. So what these secretions does is it causes the plant cells to grow excessively and form a ball-like structure. So in the diagram, you will see that there's a gull fly. So they gave you the adult gull fly and then a stem, and they showed you guys then the little ball structure that forms. So obviously the larva is found inside of this ball structure, because what the larva does is it secretes it secretes a secretion that causes these plant cells then to grow excessively. So these are then called galls. So if they talk about a gall, there's an example in the diagram on your right hand side next to the gull fly. So then they say that is called galls, which are high in nutrients. So predator birds feed on the larvae in the galls. So in that ball-like structure, you will find the larvae, and then predators will feed on the larvae. So the size of the galls produced actually depends on the genetic variation in the gull fly. So the diagram below shows the gull fly and a gull on the plant stem. So that is what we have. Let's just focus on that information. So firstly, we have the gull fly, the female that lays the eggs in the stem of a plant. Then when the larva hatches, they have a secretion that causes the plant cells to grow to form the galls, which is that ball
ball-like structure. And then predators will feed on the larva because they are very nutritious. So let's look at the summary of the investigative question. So please just note that this is not everything that you have to know. You have to please go and look at the information loaded up on the platform explaining what is needed from you, expected from you for investigative questions. So this is just in short. So firstly, the aim of the experiment is what this experiment is about. And most of the times it's given in the information at the top of the diagram or at the top of the table or the graph that's been given. So usually they give you this. So here I've added a photo of the goals where you can clearly see the stem and then the ball-like structures, which is the goals. So in this scenario, the aim is to determine if the size of the goal affects the percentage of larvae that's killed by the predators. So they want to see if the goal is bigger, will more predators target the larvae, or if the goal is smaller, will more predators target the larvae. Then we have variables. So this is the biggest problem for matriculants, and this is actually quite easy. The only difficult one is your fixed or your controlled variable. So the independent variable is what's decided, or the variable that's chosen by the scientist. And in this case, it was the size of the goal. So the size of the goal was chosen by the scientist. Then the dependent variable is what is measured. So this is something that the scientist or anyone, this is something that cannot be controlled by anyone or the scientist. So this is what is measured. So in this case, it will be the percentage of goldfly larvae that's killed by predators. And then your fixed or your controlled variable is something that's in this experiment that's kept constant or unchanged. Because as soon as you change it, it's going to influence the results of your experiment. So if I give you a table, the independent variable will always be first it will always be first in your table. So your first column will be your independent variable, which is your x-axis. Then your dependent variable will always be in the second section of the table, so the second column in your table, and that will go onto your y-axis. And this is very, very important. The fixed variable is usually also mentioned in the given scenario, but sometimes they can be quite mean, and then they ask you to mention a fixed or controlled variable that's not mentioned in the given scenario. So that's very important for you to read that specific question. So let's look at the summary of different types of variation. So what is variation? So variation is the difference of a characteristic within the same population or in the same species. So we get two different types of variation, which is discontinuous variation and continuous variation. So discontinuous variation, this is a type of variation in a population with no intermediate phenotypes. So here's a few examples. For example, eye color, gender, blood group, and then the ability to roll your tongue or not roll your tongue. Then we have continuous variation. So this is a type of variation that occurs in a population in which there is a range of intermediate phenotypes like height, foot length, hand span, or even skin color, because these traits are controlled by a polygenetic trait, or they are a polygenetic trait, which means that they are controlled by a number of genes because it shows an intermediate range of phenotypes. So let's read through the questions of question 3.4. So again, we had a female goldfly that laid her eggs in the stem of a plant. So in the right-hand corner, I gave you guys an actual photo of a goldfly in the right-hand side at the bottom, and then also the inside of the gold. So remember, the goldfly lays the eggs inside of the stem, and then the larvae, as soon as they hatch, they have a secretion that causes the plant cells to grow excessively to form a ball-like structure, which is called the gall. So if I cut through a gall, there you will see on the inside, you will see the larva in the middle of the gall. So at the bottom of the diagram,
you will see that scientists wanted to investigate whether the size of the gull had an effect on the percentage of gullfly larvae that's killed by predator birds. So then they gave you guys a table of the investigation where then you will see that there's an increase in the size of the gull. So we started at 10 millimeters, then 15, 20, 25, up until 30 millimeters in size. And then they indicated and showed how many larvae were killed in percentage by the predator birds. So at 10 millimeters, there were 1%, then 15, there was 0, 20, there was 1, 25, 2, and then 30, we had the highest percentage of larvae that was killed by the predator birds, which was 10. So in question 3.4.1, they say state the independent and dependent variable. So as I said in my summary, here you should know where to find your independent and dependent variable, or you need to know the definition. Your independent is what the scientists chose or decided on, and the independent what is measured. If you are not sure how to answer the questions like this, remember the independent is the first column and the dependent is your second column. Then they say give one advantage of the gull to the gullfly larva. So why would the secretions form the gull? So give an advantage of it. Then state why the size of the gull produced is an example of continuous variation. So before we move on to the marking guidelines, I will do a summary on what is continuous variation for you to understand what it is. So this forms part of evolution. Then explain how the percentage of gullfly larvae killed by the predators is influenced by the size of the gull. And then lastly, you have to use the given information in the table to draw a line graph. So this is very, very important. So I'll show you guys also important things in the marking guideline, which you need to look out for in the graph, and then the things that you have to be able to answer by now, because we've taught you in high school already in grade 8 how to draw a line graph, or actually all the graphs. Let's move on to the marking guidelines of question 3.4. So firstly, they asked you to state the independent and the dependent variable. So your independent variable, I said it's something that the scientists decided on, or something, the one that's first in our table, the first column. So in this case, it was the size of the gull, not just the gull. It's very important that you state the size of the gull. Then your dependent variable, it was what's measured, not just the gull larva. It is the percentage of gull larva that was killed. Very important. If you just write gullfly larva, it's wrong. So it's the percentage of gullfly larva that was killed. Then they ask you give one advantage why the gull, the advantage of the gull to the larva. They are obviously nutritious. They're there for protection, and then space. So it's a habitat, basically, a little space for them to feel safe and to feed on. Then question 3.4.3, state why the size of the gull produced is an example of continuous variation. So if you look at your table in the first column, you will see that the size increased from 10 to 30. So there was an increase. So there was an increase in the size. And if you go back to the top to the second paragraph where they say the size of the gull produced actually depends on genetic variation in the gullfly. So this is going to depend, the size of the gull is going to be affected by means of the genetics of the gullfly larva. So it depends on the secretions. So, for example, let's say the gullfly larva produces more of the secretions because of a genetic mutation or a gene. That means the gull will be bigger in size, for example. So that depends on the gullfly larva. So now they asked, how would you then know that it is continuous variation? So that means there's small changes that will be visible. So that's why you will then state that there will be a range of values for the size of the gull, or intermediate values. It didn't jump from 10 to 30 because then it will be discontinuous. It went from 10 to 15, 20, 25 to 30. So there were small changes over time.
Then explain how the percentage of gullfly larvae killed by the predators is influenced by the size of the gull. So here you have to think a little bit out of the box. So here you will state that the larvae in the 30 millimeter gulls were obviously eaten more. That's one mark. Since they were more visible, second mark, or because the bigger gull obviously contained more larvae or larger larvae. Or you can switch it around and state that the larvae in the smaller gulls, so the 25 millimeter, or the less smaller ones were eaten less. They were less visible, or there were fewer larvae in, or the larvae were obviously much smaller. So let's move over to drawing the graph. So in the graph, they give you a guideline that is used to assess the graph. So firstly, we have T. So you will notice on your question papers, there will be these letters, T, C, L, S, P. And this is how the marks are allocated and what these letters are stand for. So T is for the correct type of graph that you drew a line graph. If you drew a different graph, you will lose this mark. C is for the caption. So in the caption, you have to mention your independent and dependent variable. So if we have a look here, they say the percentage of gullfly larvae killed by the predator. So that is your dependent variable. And then due to the size of the gull, and that is going to be your independent variable. Then L is the labels for your X and your Y axis. So this one is quite difficult because you have to have four things correct. You need to have four things correct in order for you to receive this mark. So for your X axis, you need the label gull size and the unit. And then for your Y axis, you need the gullfly larva that's killed by predators. So you need the label and percentage. So you need the label and the unit. Then S is the scale. So I like using the easy multiples. In this one, it was quite easy. On your Y-axis, you use the multiples of 2. And on your X-axis, we use the multiples of 5. Then lastly is P. So here you will receive 2 marks if you have plotted correctly. So if your first 4 points are plotted correctly, you will receive 1 mark. And if all 5 are correct, you will receive both marks. So our last question for question paper 2, which is question number 3.5 regarding human evolution. So here they give you a diagram below that represents the pelvic structure. So that's number A, B, and C. And then also the ventral view of the skull of 3 organisms, which is X, Y, and Z. So please remember that the letters is extremely important. And look at the images and examine them. If we compare A to C, you will see that A is much shorter and wider. B is a little bit in between, and then C is much narrower and longer. If we look at X, you will see that the jaw is more a longer U shape, so it's a bit longer. Then Y still has canines, and then Z has a small C-shaped jaw. It's much smaller, and then you will see the foramen magnum, which is the black dot sort of in the middle of the skulls at the bottom. So you will see that X is a little bit not really in the middle, but almost. Y, the foramen magnum, is at the back of the skull, and Z is in the middle of the skull. So let's have a look at the summary of the differences between African apes versus humans. So the first characteristic, we will look at the cranium. So humans or Homo sapiens will have a larger cranium as we have a larger brain, and African apes will have a smaller cranium as they have a smaller brain. The brow ridge. So obviously humans, our brow ridges are not very well developed, where African apes have a very well developed brow ridge. The spine, humans have an S-shaped spine. It's a bit more curved in comparison to the African apes that has a C-shaped curve. The pelvic girdle for humans are shorter and much wider. So this is important for the questions that we are about to answer. And the African apes are much longer and narrower. Then canines, humans do have canines, but very small, where African apes have very large and long canines. So the palate plate, we are very small and a semicircular C-shaped, and then they also have, the African apes have a very long rectangular jaw shape, or a palate shape, sorry. Then the jaw is much smaller in humans and less protruding, where in African apes they're very large and very protruding. Cranial ridges, so humans do not have any cranial ridges, where African apes do have cranial ridges on the top of their cranium. And then the foramen magnum, also important for our question, is positioned forward to the middle in humans and then at the back for African apes. The reason why our foramen magnum is in the middle is so that we can walk on 2 feet, so we are bipedalism. So that means we can walk on 2 feet, so it's so that we can maintain balance and walk up straight. Where with the foramen magnum of the African apes are located to the back, so that they can walk on 4 feet and still maintain their balance. So let's read through the questions. So we're going to look at the pelvic structure of 3 organisms, and then also we're going to look at the skull of the 3 organisms. Please note that it doesn't mean that if we look at A, the pelvic girdle belongs to X. It is random, so it doesn't mean that the organisms belong to one another. So they say write down the letter or letters of the diagram that represents the skull of bipedal organisms. So this means bipedal means walking on 2 feet. So they said letter or letters. So that means it can be more than 2 letters. So they said the skull. So it doesn't help that you write A, B, or C. So you have to look at X, Y, and Z, and it's 2 marks. So it's easily seen that you have to write down 2 organisms. Then the pelvic structure of an organism that is quadrupedal. So this is an organism that walks on 4 legs. Then give a reason for your answer in question 3.5.1b. Then describe one other structural difference between a bipedal and a quadrupedal organism. So describe one structural difference between them. So let's look at the marking guidelines. So why is bipedalism so important? Is that we have hands. Our hands become free for carrying food, tools, and babies. We can move more efficient. We have a better view of our surrounding. Our bodies can also cool down faster, and it also allows us to it allows the display of the male sex organs for courtship behavior. So then, the marking guidelines question 3.5.1 say that they want letters or one letter or letters that represents the skull of an organism that walks on 2 legs. So that will be X and Z. So it can be in any order. So it's only X and Z because of the location of the foramen magnum. So if it's located to the middle of the skull, it's at the bottom, so if it's located to the middle, they will be able to walk on 2 feet. Y is not one of them because the foramen magnum is located at the back. Then they ask you the pelvic girdle of an organism that walks on 4 legs, so that will be C. Please do not say X, Y, or Z. In this case, it is C because they asked the pelvic girdle. And then in question 3.5.2, they ask you a reason, and then you just describe the structure that you see there. So you will see that the pelvic girdle is long for 1 mark and also narrow for a 2nd mark. So please just note that they might ask only one. It can only be 1 mark, but please make sure that you always write long and narrow together. 3.5.3, describe one other structural difference between an organism that walks on 2 legs and an organism that walks on 4. So the other structural differences is the spine. The spine. So the spine is S-shaped if you walk on 2 feet, and it's C-shaped if you walk on 4 feet. Or then also the foramen magnum. Here you will see that if you walk on 2 feet, it's located to the middle or to the front of the skeleton, or to the skull, sorry. And then if you walk on 4 feet, it will be located to the back of the skull, and I mark the first 4. So you just speak about what the structural difference that's mentioned in the diagram. So not any other structural differences. So they want to see what is asked in this section. Thank you, grade 12s. Please make sure to watch the next few videos on the question papers.